Can't wait.
Great, thank you. Yeah, so as Sergio mentioned, my name is Mallory Embree. I'm the co-founder and chief science officer of Ascus Biosciences. My background is I have a PhD in bioengineering from UC San Diego. While I was there, I studied microbial communities. So I'm not actually an animal scientist, but I know a lot about microbes and how they interact. And I basically dedicated myself to find new ways to study how these microbial communities form and how to elucidate what these microbes are actually doing. So in this presentation, I'm going to spend majority of the time just talking about how we actually study the complex microbial communities to create the products that we're now selling. So with that, so one of the things that Mike, the CEO of Ascus, and I first noticed when we created Ascus about 5 years ago or so now, was that animals, livestock animals, actually receive a lot of microbes in their feed already. When we started to look at what microbes were actually in these products though, we realized that it was actually a very short list. So it turns out that 90% of the actively marketed DFMs are actually comprised of the same 4 microorganisms that you can see on this chart over to the right here. Just to point out a few more, there of course is Saccharomyces cerevisiae, which is well known for its use in bread making as well as beer fermenting. Lactobacillus acidophilus, which is actually a human intestine microbe that's used in yogurt making. There's also Bacillus subtilis, which is a very common soil microorganism. Like if you were to go outside and sequence some of the dirt anywhere, you'll find some Bacillus subtilis. There's also Enterococcus faecium, which is another human intestine microorganism. And then Aspergillus oryzae, which is used to make soy sauce, miso, and sake. And so there's nothing necessarily wrong with these organisms, right? They were discovered decades ago, primarily because they could grow very, very well on agar plates, right? Those little petri dishes that you see in microbiology labs a lot. And it's very, very easy to grow them to scale just because they're very, very good at growing in flasks under these man-made conditions. Over the past 20 years or so, though, we've really learned a lot about the microbiome, like first that it even exists, right? And many, many people in the scientific community have really dedicated themselves to understand what these microbes actually do inside their hosts, right? And so if you look at the microbiome versus these microbes, this list of microorganisms, they're considered ecological outsiders. So they're not actually native to the animals that they're actually being fed to. And so that's really why we decided to start this company, because we think that better products could be made that are actually derived from native microorganisms. And so now, if you look at why native microbes are better, there's plenty of reasons why, and I've just kind of boiled it down to 3 here. The first is that they actually have genes that enable them to thrive in their host environment. So if you look at rumen microbes, right, they actually have the genetic content, they have the genes that allow them to utilize TMR effectively for both themselves as well as their host animal. There's also a very long list of host factors as well here. These tend to be different signaling molecules, carbohydrates, proteins that are actually present on the surface of the microbes themselves, their membranes, right? These molecules enable the rumen microbes to actually recognize the rumen environment as their home. Like they get there and they realize like, okay, this is where I'm supposed to be growing. This is my niche. This is my community. This is where all my friends live. And the vice versa is actually also true as well. So because the microbes have all of these special things on their membrane, the cow rumen epithelial cells themselves are actually able to recognize the microbes as a friend, right? They recognize that it's actually a native microbe that's supposed to be there, as opposed to a pathogen or something that might actually be deleterious to the host. And then finally, and this is the part that I really, really like to study, these native microbes actually have the ability to interact with the entire community. So microbes are very, very good at identifying who their friends actually are. They know who they're supposed to be in a community with. And they have many tools available to them that actually lets them suppress some of these outsiders, right? Like antibiotics, for example, are a way that community members can actually suppress microbes that they don't necessarily want to be in the community with them. And so that said, right, it sounds great, right? Everybody wants to go and tackle the microbiome and find these microbes and ways to manipulate it. But it turns out that it's a very challenging system to work with just because there's tens of thousands of different microorganisms present in this complex environment. So on the left here, we just have an example of some of the rumen samples that we actually sequenced several years ago. Each little bar here represents a different species, right? So not even at the strain level, just at the species level. And so you can see that it's incredibly complex. There's many, many things living there. And it's not a trivial task to actually go in and pick out just a handful of microorganisms to turn into a product. And so that's where our platform comes into play, right? And so this is some methodology that we've invented to enable us to go through these tens of thousands of microorganisms and actually pick out the most efficacious ones for products. And so in each case, right, this is the workflow that we follow to actually generate our target list. We always do everything de novo. So in the case of Galaxis, our dairy cow product, we started off by essentially surveying hundreds of cows. We literally went to all of these cows that were on commercial farms and academic trials with CROs, and we took their rumen content. Once we had the rumen content, we went in and isolated the nucleic acids from the different microbes that live there, and we sequenced them using next-gen sequencing. This generates a lot of data, and I'm gonna go through each one of these steps in a little bit more detail. This is just kind of to give you a sense of what's coming. So once we have the sequencing data, we then integrate additional data sets to give us a higher resolution of what's actually happening in the microbial community at that time. These data sets are essentially cell count data. So we get a sense of how the microbial biomass is changing over time, as well as transcriptomics and animal metadata. So how much milk was that cow making that day when we isolated the rumen content. From there, we apply a lot of mathematical methods to actually explore the different relationships between the microbes themselves, as well as the microbes and the metadata. And from there, we generate a network that allows us to identify the top targets that we want to isolate from the rumen content to turn into products. So to get into a little bit more detail then, I guess to start off, I guess this picture should be very familiar to all of you, right? This is essentially a very high level of the digestion in the rumen that's mediated by the different microbial communities. Essentially, you have all your macromolecules up here, your cellulose, your starch, your protein, your lipids, that eventually gets fermented down into different volatile fatty acids. In some cases, you'll make methane, for example, CO2, hydrogen as well. And so when we started off with ASCUS, we were really, really excited about looking at dairy cows because essentially we already knew that as a scientific community, right, we already knew that the rumen is very, very important in helping the animals actually obtain the nutrients and energy from feed. And so it was a really good test for the platform to see if it could actually pick out some of the beneficial organisms that did these processes better. And so here is a high level overview of what the Discovery platform is. So there's two aspects, right? The microbiome aspect as well as the animal aspect, because again, we're doing these survey experiments at the very beginning where we're taking samples, microbiome samples from the animal to understand how they link to the phenotype of the animal. To kind of go into these first two aspects here first, this weighting community populations and determining active microbes. The way you want to think about these two steps is we are essentially just getting a more accurate picture of what the microbiome is doing at that particular moment in time when the animal is sampled. And so to start off, here's a little bit of math for you guys. So it turns out that microbiome data sets are something called compositional. So that means that typically when you take your sample and you put it through the sequencer and you generate the reads, what comes out on the end is actually a percentage. And so if you were to pick up any sort of article on the microbiome or any sort of academic article particularly, and read through the results, oftentimes the results are reported in percentages, right? In sample 1, we saw 50% lactobacillus. In sample 2, we saw 10%. And there's not necessarily anything wrong with looking at the data in this way. The issue is when you try to compare across conditions. So in this example here, you have your bacterial population, you sequence it, what you get out is this proportion of bacteria, so a percentage. And so case 1 here is pretty self-explanatory, right? It looks like you have less A than B, maybe 25% A, we can say. The issue comes when you try to compare it to the second or third state. So the percentage, right, if you're just to compare the abundance of A across conditions 1, 2, and 3, it looks like A increased in conditions 2 and 3. The reality of the situation though, right, is that these microbes actually exist as an absolute number of cells within these samples. And so it might turn out that condition 2 actually has less cells than condition 1, and maybe condition 3 has the exact same number of cells as condition 1. And so if you're actually looking at how A changed in conditions 2 and 3 as compared to 1, because condition 2 has less cells, it turns out that A didn't actually change. Since 3 had the same amount of cells, it did actually increase. And so you can see that you can sometimes come to false conclusions if you only look at the percentage that's being represented by these data sets. There's also a lot of issues with the math itself and applying normal statistics to these. Correlation, for example, is something that people really like to apply to these data sets, but since these matrices tend to be so sparse, meaning there's a lot of zeros with them, you sometimes generate false positives, right? You get these spurious correlations that kind of take you down the wrong path. And so to address this, instead of looking at everything as a proportion, we actually quantify the number of microbes in each sample before we actually analyze it. And so we have a flow cytometer in our lab that actually allows us to count the different number of bacteria and fungi in each sample that we sequence. And this enables us to get around that, that limitation with the percentage-based analysis. And it gives us a more accurate reading of how the microbiome is changing over time. The other thing that we do is we use transcriptomics to actually identify active microorganisms. And so if you look at the central dogma of biology, there's many different places you can measure to actually figure out what microbes are on and actively doing something in the community at that point in time. So you can look at things like DNA replication, for example. I'll just spoil it now. We look at RNA to actually measure activity. But you can also look at the protein level or the metabolic level with either proteomics or metabolomics. Transcriptomics is fairly straightforward and cost effective, and so that's why we decided to go that particular way. We do a little bit with ribosome profiling, but again, it's still a little expensive at this point, but it's pretty cool. And so that's kind of the wet lab aspects of our discovery platform. So once we have those pieces of information, we have a fairly comprehensive view of what the microbiome looks like at that given point in time. And so from there, we go into more in silico computational methods to actually pick out the microbes that actually matter. And so remember, when we do these survey experiments too, I guess one thing to point out is that they always tend to be longitudinal. So that means that we don't just take one sample from a cow. We actually follow that cow for quite some time, It depends on the experiment, but sometimes it was for a month, sometimes it was for a couple months. And we would take multiple samples from that animal over time just to see how it was changing. And the idea here is once you have this time series abundance, a lot more mathematical tools actually open up to you. The ones that we're most interested in are these interaction networks that we can then boil down to basically different modules or clicks. And I'll go into a little bit more how we do that in the next couple slides, but the goal of this, right, is to take that really, really complicated community that has tens of thousands of different species in it and actually collapse it down into something that's a lot more interpretable, that we can actually relate to function. Because at the end of the day, you're most interested in what these microbes are actually doing for the animal. You're most interested in how they interact with each other. And it's really this assignment of structure that enables us to do that. And so at the end of all of this, right, you have all of these organisms that are classified into these different cliques, right? And so these clusters of cliques we found actually represent organisms with similar function, or they're organisms that are in a very tight symbiosis or syntrophy where their metabolisms are very, very linked together. And so, of course, there's many ways to mathematically model some of these complex microbial communities. You can use non-parametric models. There's machine learning algorithms that you can use, as well as some graph partitioning. If you guys are curious, I'm more than happy to talk about this stuff, but for the sake of time, we'll just talk about the one that we actually use. So the particular mathematical method that we use to actually generate these interaction networks is something called mutual information. And so the way this works, so remember we have our longitudinal data set again, where we have several points for the animals over time, along with this microbiome data. Essentially, you lay that all out in a plane, right? And you stack it along with all of the different cows that you've measured, right? And you essentially look at the probability distribution of these particular curves, and you use a computational method called meshing to basically calculate out the mutual information coefficient itself. And so, I know it's a lot of words, it's a lot of math, but all you really need to know is if these points tend to line up particular ways in the mesh, the mutual information coefficient basically assigns a numerical value to it from 0 to 1, where 0 means that there's no relationship that was detected in the mesh, and 1 means that there's a very strong relationship that was detected. So once you have this mesh of interactions completely mapped out, we then apply some network topology tools to actually figure out how these microbes are grouping together. And so you can kind of see here on the left, you have a network that hasn't been optimized. Essentially, you run, we use a particular algorithm called the Louvain partitioning algorithm, and it uses these linkages between nodes to find the minimum clustering of the network. And so it's something that kind of iterates over and over and over. Every single time this algorithm runs, more and more edges are collapsed until you end up with a very simplified structure assigned to your network. So you can see here, you go from something very complicated. From the first pass, all of these nodes are assigned to the individual modules, and it continues to be iterated through first pass, second pass, as many passes are needed until it's really collapsed into the bare minimum module structure of the entire network. And so what do we end up with at the end of this, right? Like, you don't need to understand how it works. That's just what we do. What you end up with is a network that looks something like this, where each node, so each circle, represents both microbes as well as animal metadata. And the edges between these nodes represent the different interactions that we detected using the MIC, using the meshing. And again, the whole goal of doing this is to make the tens of thousands of species that live within the community into something more digestible, so we can actually pick out microbes to turn into products. And so basically what you need to understand from this is that organisms that actually perform similar chemistry or are so tightly bound through syntrophy that they essentially share some sort of metabolic chemistry fall into the same cluster, right? So things in this particular orange bubble will behave or will be doing similar metabolic things to other members of that cluster. And so when we go in and pick products then, we tend to pick strains from different clusters to make sure that we get different metabolic capabilities that are actually represented in the product. So for example, you don't want to pick two butyrate producers, right? You'd rather have a butyrate producer and a fiber degrader, and so you'd pick from different bubbles to maximize the effect of the product. And so going back a little bit, so that was just how the platform works. So if you guys have any questions about that, please ask. Now for Galaxis specifically, this is just a description of some of the different characteristics that we looked at when we actually did the survey experiments in the beginning of the study. Because remember, when we create products, it all starts from de novo research. So we do all of this intensive sampling to understand what the microbial community looks like under various conditions. And so some of the conditions that we looked at during the survey experiment were breed. So we sequenced both Jersey as well as Holstein cows. We looked at different feed regimes. So we looked at both PMR and TMR. We looked at different levels of forage. We had a couple milk fat depression experiments where we actually took animals and we increased the amount of corn in their diet to induce a change in the milk production. We looked at various different environments, right? Because universities are very different than a commercial farm. We took samples across different seasons under different weather conditions. And so we looked at a lot of different things. And potentially the most important thing here, and we actually get this question a lot, is it's about the core microbiome. So it turns out that there is a subset of microorganisms that live in the rumen that are shared across pretty much every single cow. And so this is something we found early on in our survey, but academics have also begun to realize this too. Like there's some pretty cool Nature papers that came out, I think it was 2 years ago or so, where they sequenced cows from all across the world and they identified the same core microbiome too. So it was very important that we not only pick efficacious microbes, we had to pick microbes from this core rumen microbiome as well, just to make sure that the product would work in every single cow. Like I'm sure there's gonna be some cows somewhere, maybe it's like a mixed breed or something, and it's just totally special and it has a completely different microbiome that falls out of the norm. But if you take your typical Holstein or Jersey or whatever relevant breed you're looking at, there's a 99% chance that it also has this core rumen microbiome. And so that's how we essentially made sure that the microbes had the highest chance possible of working in every single cow. All right, so now let's get into the microbiology a little bit. So remember from the platform, we end up with this target list, right? It's just a list of sequences of the most wanted microbes that we want to turn into a product. And so I guess the struggle here is that there's actually a lot of microbial diversity that's very, very unknown. And it's starting to become better now that we can actually do sequencing. But if you look at rumen microbiology up until about the early 2000s or so, most of it was actually agar plate based. And so this kind of goes back to the first slide I showed too, where most DFMs tend to be these organisms that are easy to grow, right? Like the soil microbe, for example, you could just take any dirt, put it on a plate, and I guarantee you're gonna get a Bacillus subtilis out. And there's nothing wrong with that. That's just where the technology was. It's just gotten a lot better. And so now we're starting to really understand the microbial diversity that actually lives in the rumen. And we're starting to understand how the community works better and how it links to physiology. If you look back to the early days of Ascus though, in 2015, when we were first isolating some of these target microorganisms, most of the things that we were going after didn't even have a taxonomic name assigned, which is pretty crazy. So of course, if they have no name, that means nobody's ever studied them before. And so we've had to do a lot of very basic microbiology characterization on these strains, even before we kind of took them up the process development chain to turn them into products, just because we didn't know how to grow them. Nobody had studied them before. More recently, fortunately, there's other academics that have begun to really address this gap in knowledge. And so more of these organisms are getting names. But still, it turns out that sequencing can only take you so far, right? Like sequencing the genome of an organism can kind of tell you what you think it should be doing from a metabolic level and the genes that it has. So the sequencing, I guess what I'm trying to say, is very, very different from actually culturing microbes. It turns out that isolating different microbes from rumen content can actually be very, very challenging. And so we have a lot of tricks and tools in our lab that we use to basically up the probability of getting the microbes that we actually want from these samples. And so at this point, I think we have over 300 unique species in our library. So when I say unique species, that means taxonomically distinct. So we don't have a library of 3,000 Saccharomyces cerevisiae. We have well over 300 different rumen species present in our freezers here. And so we have lots of microbes to choose from for future products. But yeah, right now we just have the 2 that we are focusing on. So yeah, we have a lot of microbiologists here at ASCUS that essentially spend all of their time culturing from animal GI samples. And so one really interesting thing that we started to see once we actually started pulling out these isolates and characterizing them is the way they bias their carbon utilization between catabolism as well as biomass generation. And so what I mean by that is, imagine you're a microbe, you're in the rumen, and a great glucose molecule shows up, right? Everything in the rumen really, really loves eating glucose. So you take that up into your cell and you have 2 choices of how to use that glucose molecule. So first, you can kind of send it down these catabolic pathways. You can ferment it. You can use respiration, right? You have all of these choices if you decide to pass it down just your core metabolic routes to generate ATP and byproducts for the cow. The other way this carbon can go is actually to generating biomass. So this essentially means building up the different core building blocks within the cells. So all of the different nucleotides, the amino acids, the lipids, the other carbohydrates, so that you can multiply your cell. And both of these have their benefits for the animal, of course. You don't want to bias completely one way, for example, because both are necessary to create a healthy cow. Biomass, of course, goes to protein for the animal, and the byproducts from catabolism generate the energy that the animal actually needs to grow and be healthy. What we started to find when we pulled out all of these microbes from these samples is that majority of the top target strains that we were interested in were actually more efficient at the catabolic processes. So they were the ones that biased towards catabolism rather than biomass. And so what we have here is something called a rank abundance plot. And so essentially we figured out what the abundance in relative abundance terms was for each one of these microbes. It goes from 0 to 100. And what you should see is that it's very, very skewed. So there's a couple microbes that make up the vast majority of the community. But then there's this huge tail of microorganisms that goes out that are all less than 1% or so. And it turns out that, of course, the strains that we're interested in are all less than 1% for the most part. And so again, these microbes are the ones that are spending, I guess, their metabolic powers to actually convert the TMR into valuable molecules rather than just growing to a high cell density. And so the other thing that we do once we actually have these isolates in pure culture is we genome sequence them. We generate the whole genome sequence of each one of these organisms to basically gain insight into what these organisms might actually be doing within the rumen. So here, for example, is dairy 20, which is the bacteria that's present in Galaxis. And from the genomic information, we are able to determine that it sporulates, which is great for process development and figuring out stability of the strain. You can look at things like just carbohydrate metabolism, for example. You could look for antibiotic resistances, and you could try and figure out any other, I guess, cool genes that might make it special. Once we have the genome sequence, we actually study them in vitro as well to just try and confirm some of the things that we detect in the genome. And so once we've done that for all of the strains that we're interested in, right, we go back to the initial network analysis that we did to try and further define how these different microbes are actually interacting with the cow. So for example, we found, because remember these nodes represent strains as well as animal metadata. So this blue bubble here represents milk fat in this case, because this is where the milk fat metadata tended to fall. When we started looking at all of these individual microorganisms and what they were potentially doing in the cow, we realized that all of them were actually butyrate producers. And so we found literature, because again, we weren't animal scientists, right? We didn't have any animal scientists on our team at the time. We were a team of microbiologists, molecular biologists, and bioinformaticians, right? We were able to find these manuscripts that essentially showed that when butyrate was directly infused into the rumen, milk fat increased. So that was very interesting for us. Once we finally have the strains, you can do a lot with them to basically try and understand how they actually impact the cow. All right, and so the next thing I want to touch on is, again, this idea of microbial cliques and why they're important. And so when researchers tend to look at the rumen, right, it's really easy to look at it in terms of linear pathways. For example, right, you have pyruvate, it can be broken down into acetyl-CoA, lactate, succinate, formate, and H2, right? It's very straightforward. It's just how we know the chemistry works. It's there, and we just know that it proceeds at different efficiencies in different animals. So the reality, though, is that these pathways, right, are actually done by the different microbes, and these microbes are actually governed by the different social structure that exists between them. So remember when I was saying earlier that microbes are very good at identifying their friends within the community, right? Friends will get advantages that the unfriended will not have. And so it's some of these more subtle interactions that actually drive the efficiency of different processes within the rumen. And so it turns out, of course, that some of these cliques of microorganisms are more efficient than others. And so I know that if you've seen some of our presentations before, we always like to show that lower species diversity means more efficient production because there's less competition. But this other example is something that I think is really interesting that I want to show. Essentially, right, this figure details out two different competing pathways for pyruvate. So in the first case, right, you have pyruvate and you might have a particular clique that ultimately turns it into methane. And methane generation, of course, is not the greatest because the cow will just belch it out and it's lost carbon to the environment. And since it's not being assimilated, it's essentially a waste of feed. You can have a different clique take the same molecules and potentially turn it into propionate, which of course is a much better outcome for the cow. And so how does the cow bias towards one or the other, right? It turns out that the rumen microbiome is very dynamic, but it's heavily influenced by environmental as well as physiological cues. And it's these cues that encourage some of these more dramatic dysbiotic events in the rumen itself that allows for different groups of microbes to actually take over and skew the chemistry of the rumen in one particular manner. And so without these perturbations, it turns out that transition into these different spaces is a lot more difficult. But sometimes, depending on what the state actually is, it can be an easier transition if you know what the cue actually is. And so this matters, right? Because of Galaxis and the way that we're telling people to use it, right? And so when you actually feed Galaxis, you're increasing the abundance of rumen microbes. So in this case, it's dairy 20, the bacteria, as well as dairy 21, the fungus. And we've confirmed in our experiments that when you feed it, right, you're essentially increasing the abundance of dairy 20 from anywhere from 0.01 to 0.001%. You're taking that low value and increasing it up to anywhere from 0.1 to 5%. For the fungus, you're taking it from 0.1 to 1% up to 1 to 10%, which doesn't sound like a lot, right? But it's actually fairly substantial. The important thing, though, right, is that these are cornerstone microbes. So even though you're only affecting these two microbes, right, and I've overlaid them in the areas where we believe they're acting, right, we believe dairy 21 is helping break down some of the larger macromolecules, including cellulose and starches. Dairy 20 is making butyrate as well as other volatile fatty acids at a much more efficient and rapid pace. So by increasing the abundance of these microorganisms, you're actually increasing the abundance of their clique as well. And so in that manner, you're skewing the chemistry that's actually happening within the rumen. And so it turns out in our experiments, we've basically measured that these two microorganisms, even though you're only increasing them a little bit, right? Because, well, yeah, so even though you're only increasing them a little bit, it turns out that you're actually impacting over 20% of the rumen microbial community. So you're increasing the abundance of all of their friends too by increasing the abundance of these cornerstone microbes. And if you look at this in terms of transitioning states, it turns out that daily feeding of these microbes acts almost like an environmental cue. And so it's this feeding that actually encourages the shift of the microbial cliques to a more efficient state. And again, this all kind of links back to that network analysis that we did at the very beginning, because if you understand how all the microbes are connected to each other, as well as the metadata, you can pick out the right ones to cause the greatest impact in the rumen itself. And so the last thing I want to touch on is this idea of microbial colonization. So Galaxis, we have spent a lot of time figuring out how to actually deliver live microorganisms to dairy cows, right? And by that I mean, we're trying to find ways to basically deliver these anaerobic, sometimes vegetative microbes in a bag through TMR, and then have the TMR sit out for a while until the cow actually ingests it, right? And it turns out that feeding live microorganisms is actually really, really important. And so if you only feed dead cells, right? So it turns out a lot of the DFMs that are available today actually are not necessarily as viable as they say. And so if you feed dead cells, for example, let's just pretend the dose is 1E8 cells per head per day. These cells get into the rumen, which is a very, very big place, and then nothing happens, right? They just kind of dilute themselves out so that you end up ultimately just with 1.36 E6 cells per liter, right? And so that's a very small number of cells. Maybe it'll do something, it's hard to say. But when you feed live cells though, you add that same 1E8 cells per head per day, it gets diluted to the same value, but since they're actually alive, they can actually start doubling. So microbes like to grow exponentially. And so if you apply some of the growth rates that we see in our lab, of course, tailored down because it's a competitive environment, after 24 hours, this 1.3E6 cells will actually substantially increase to about 9.2E8 cells. And so you can see that it's really, really important to make sure that Galaxis is actually delivered alive to the rumen, because you need this colonization right? You need these cells to actually start doubling so that they can have that trickle down effect where they actually start growing and they can influence the different cliques that they're a part of. And I think that's all I have, Sergio. That's all I was going to cover today.
Great.
So yeah, if you guys have any questions, let me know.
¿Alguno tiene alguna pregunta, algo para comentar con Mallory, alguna cosa para aclarar?
A ver, yo me pongo a pensar, ¿sí me escuchan?
Sí.
Sí.
Y hay diferentes sistemas de manejo, muchos en pastoreo, muchos en los establos al estilo americano con gran cantidad de carbohidratos. ¿Harían o hay producto que va más enfocado a esa dieta o todo es general?
¿Te referís a tener distintos tipos de productos dirigidos para un tipo de alimentación determinado?
Sí, o sea, que en su bolsa venga etiqueta para, vamos a decir, establos con alta producción y sin pastoreo, con niveles de granos altos, por decir. ¿Traerían diferente dichos?
Okay. So Mallory, what he's asking is if you think that we should have different targeted products like Galaxies or Altius for more grain-based rations and for more pasture-based or fiber-based rations or TMR, PMR based on the specificity of the microbes.
Got it. So it depends on how wide of a range we're talking here. So one of the things that we have under development is a beef feedlot product, and that's intended for cattle that are on 90% grain. And so I guess my point is if you're feeding dairy cattle closer to that range, there's definitely other chemistries that you need to be aware of. For the other direction, right, lower concentrates and more forage, Galaxis is meant for that sort of case, right? You have dairy 21, that's your cellulose degrader, and it'll work no matter what the forage concentration actually is. It's just when you get to that higher end of concentrate, the animal's under very different stresses.
Correcto.
Pero bueno, Jorge, contestando tu pregunta, soy Alejandro Chávez. Prácticamente nada más tenemos un producto que combina estos dos microorganismos y que actúan hasta ahora bien en cualquier tipo de dieta o pues sí, básicamente de dieta. Entonces, esa es la respuesta concreta a tu pregunta. No hay más que un solo producto. No hemos hecho ninguna adaptación a diferentes dietas y eso. Estamos usando el mismo producto, básicamente.
Correcto, gracias.
Mallory, I also have a question. When you were speaking about the discovery, you showed the network graphics. So that could be related like social network when you map all the interaction between the individuals. And we made sure that we picked a microbe for every activity region and every network section.
Exactly. So a lot of the math that we utilized here actually came about because of social networks becoming big things, right? Like Facebook and Instagram and all of that, just because those developers are really interested in seeing, I guess, how people interact and how they respond to different cues as well, right? Advertising, for example. And so microbes have a similar social structure to people in that sense. And so those algorithms actually applied very, very nicely to the microbial community analysis that we did here. But yeah, ultimately, when we cultivate our microbes, we do try to go for each one of those bubbles so that we can get a better sense of the function that's being described by it. And so there's, of course, some that we can't get just because those particular microbes are very difficult to culture in a lab setting, right? Not all microbes are readily culturable. And so, yeah, we do get the vast majority of the things that we want, though. And that enables us to kind of pick and choose which ones go into the product.
Good. I also have another question from the audience, and they ask if you need to feed galaxies with the TMR, or it permanently, or if it will shift and change the rumen populations in a permanent way that stabilizes, and you don't need to continue administering with the TMR.
Mm-hmm. And so we think that it has to be fed every single day in order to maintain the perturbation. And so there's some really nice academic papers that came out where they actually swapped the rumen content of two cows. And they found that after a month, the host factors of the cow forced the rumen back to the original state. And that's something that we see in our data set too. Like, you still have these host factors that are trying to shape the microbial composition. And so you need to feed it every day to induce that environmental cue to keep it in that more efficient state.
Good.
So, hello, I have a question. Can you hear me?
Yes.
Okay, thank you. Yeah, my name is Agustin Delgado. I am pretty much doing nutrition in the area of Chihuahua and Texas and New Mexico. And thinking about this kind of products, one of the questions that I brought to my mind is that what is the minimum dose that you need to use for this product, and also the maximum dose that you can use in a regular basis with your herd to obtain the better performance of the cows? And the other question is going to be pretty much linked to the other question that you had before. Is now that we know the different kind of digestibilities that we have in the forages, when we will expect to have the better response by using this kind of product, understanding that we have a different variabilities on the forage digestibility in the diets? It's pretty much linked to the first question.
Okay, so back to the minimum and maximum dose. So we actually started off some of our initial trials with more cells than what's currently being included in the bag. And we found that when more cells are fed, you actually see a response slightly faster, which makes sense because if you're feeding more, that environmental perturbation is greater. And so it doesn't take as long for the cells to kind of hit that threshold where they need to be to actually induce the changes in the clicks that we see. Minimum, we haven't tested much lower than what's available in the bag already, but theoretically, right, if you follow the same logic, you could feed less, but it would take a lot longer to actually see a response. And then you're also fighting with that particular level not being as much of an environmental cue, right? Like, you might not actually get to the point where the microbes are able to grow up to the level that I showed in that one slide. Where did it go? Here. Right, so we haven't tested the minimum aspect of it, so I can't give you a cell number, unfortunately. But maximum, well, you can't overdose the cows with these microbes. There's no way, just because the community itself will actually suppress microbes if they start to get too high in abundance. So you will not be able to see dairy 20 above this 5% level, just like you would not see dairy 21 above the 10% level, just because the natural community dynamics will not let a single microorganism take over. So I think that was your first question. And then your second question was about how does it impact different forage digestibility, so different quality of forage?
Yes.
So we haven't done extensive work on that yet. So I can't tell you if Dairy 21 will digest a poor forage better than like a different microbe, for example. On some in vitro tests that we've done in our lab, it can break down pretty much anything. Like we can actually grow it on paper, like cellulosic paper. So yeah, I'm sorry, I can't answer that question. We haven't tested it in that particular condition in cows yet.
All right. Thank you very much.
Mm-hmm.
Y bueno, solo Agustín, una respuesta práctica a tu pregunta. La dosificación por vaca es de 5 gramos por día y eso ya incluye el mínimo requisito que estaba mencionando Mallory. Entonces, 5 gramos por día es una respuesta práctica ya del producto.
Perfecto. La pregunta era también la parte de la digestibilidad, porque uno de los aspectos que se ha estado investigando ya de unos 3, 4 años hacia atrás, hacia la fecha, es que se trata de entender un poco más el metabolismo para la producción de grasa en la leche. Entonces, uno de los factores que afecta es la síntesis de novo de esos ácidos grasos, que básicamente proviene de acetato y del butirato. Entonces, a ese lado iba encaminada mi pregunta, para ver si tenían un poco más de información en ese sentido.
Todavía no, Agustín. Estamos trabajando para diseñar esos estudios en diferentes tipos de fibras, diferentes formulaciones, y todavía no tenemos bien los datos, pero lo que contesta Mallory es interesante, de que realmente ha funcionado bien en todo tipo de elementos. Pero sí creo que es importante, Agustín, y gracias por tu pregunta, enfocarnos en diferentes tipos de ingredientes y de granos y de fibras para tener una idea más amplia de en qué condiciones puede trabajar mejor el producto. Entonces, estamos diseñando esos estudios y bueno, esperemos pronto tener un poco más de información al respecto.
Enterado, gracias.
Solo para aclarar la confusión, si no lo han visto en el chat, la dosis es 5 gramos por vaca por día para Altius en México, 50 gramos por vaca por día para Galaxis en el resto de los mercados. Tienen exactamente la misma concentración de Dairy 20 y Dairy 21, los dos endomicrobios que están presentes. Simplemente una cuestión de manejo y dosificación. We have another question for you, Mallory, following up of what he asked before. What's the partition or the percentage in the increase in butyrate, acetate, and propionate production?
Mm-hmm. Yeah, I can't give those numbers, unfortunately, either. So that's not something that we've measured commercially yet.
And Mallory, this is Alex. Just for the benefit of the group, could you just let us know where are we headed in terms of research and the new studies that you are doing with other universities? Just in general, where are we headed in backing up the product with more research? And so if you just can give us an idea for the benefit of the whole group.
So over the next 6 months or so, we're going to be starting a number of trials up here in the United States with academics at a variety of universities, including Cornell, University of Illinois, Michigan, South Dakota State, where we're going to be testing Galaxis for a very long period of time. Essentially, we're going to start feeding them Galaxis early in lactation and run an experiment for at least half a year to see how it impacts the animal across the entire lactation curve. We'll be testing Galaxis, and we're actually going to be testing the next version of Galaxis too. The second version of Galaxis contains the same 2 microorganisms in the first version of Galaxis, plus another 2 novel rumen microorganisms that again, fall into the different bubbles in that click. And so we'll be generating a lot of data over the next couple months so that we get a better idea of how both of these products impact the animal over a longer period of time.
Thank you, Mallory.
Also, Mallory, it's important to mention that we are also working with other animals. You already mentioned that you are working with beef cattle, but also with other animals, and soon we could have in the market a product for poultry, for swines and pets and other animals. No?
Exactly. So we're working on a broiler chicken product where it essentially is a collection of microorganisms that prevent necrotic enteritis from infecting the birds, so they have healthier GI tracts. We're creating a beef feedlot product, so specifically for beef on a feedlot high grain diet. And there we're seeing about 10% improvement in feed efficiency. We're also working on a horse product for colic, and then a dog product as well for just general GI health.
Mallory, Sergio, tengo aquí una pregunta de Jorge Montemayor, nada más que entró en privado. Dice, si solo damos esta cantidad por vaca, debemos premezclar en los minerales para lograr que se reparta bien en un carro mezclador. ¿Es estable y se puede hacer? No sé si lo haces a Mallory o contesto yo por acá.
Contestarle directamente entonces, ya que lo hiciste en español.
Sí, sí, doctor. Jorge, el producto es estable por 24 horas una vez que lo abrimos la bolsa. Entonces, no lo estamos mezclando directamente en las premezclas, en las plantas, por esta característica que son microorganismos vivos. Entonces, no viven más de 24 horas, lo estamos haciendo directamente en el establo, en la premezcla que hacen en el establo. Así es como lo estamos trabajando, Jorge.
Gracias.
Gracias a ti, Jorge.
Bueno, ¿alguna otra pregunta, algún otro comentario o alguna duda para Mallory?
No.
Oh, we have another question, Mallory. What's the effect that galaxies might cause in the development of the cows when using in heifers?
Heifers that are lactating or pre-lactating, or?
Heifers, yes, it's pre-lactating. It's before the first calving.
Okay, so even before they're lactating though.
Yes, so they're actually still growing.
Okay, so at the end of the day, what Galaxis is doing is releasing more energy from the feed. And so if there's something that the heifer is going to benefit from having more energy, right, better digestibility, the product should also help with that too. We've never tested that directly, so I can't tell you like feeding it to heifers will make them bigger, faster, or anything like that. But in general, we see more energy is better. So I imagine it would be beneficial for them as well.
Yes, and something we've been, okay, I will take it in Spanish, Mallory.
Sure.
Y algo que hemos estado también trabajando con profesores de fisiología acá en Argentina, de la Universidad de La Plata, es que es la vaca en definitiva quien va a decidir de acuerdo a sus necesidades a dónde va a dirigir esa energía extra que sale de un mejor funcionamiento ruminal. Entonces puede ser para mantenimiento, puede ser para fertilidad y reproducción o puede ser para producción de leche. En este caso también entraría el desarrollo, pero en definitiva, en última instancia, nosotros lo que hacemos es potenciar el rumen para que tenga un mejor funcionamiento, entregarle energía extra a la vaca y es la vaca quien va a decidir a dónde lo va a ir derivando. Mallory, you have another question based on the historical trials up to date. If we have noticed any difference in dry matter intake between the treatment and control group.
Yeah, so in the ISU trial that we did see a lower DMI in some of the cows that were already producing a lot of milk. So in those particular cows, we think that they were already making, they're almost at the physical maximum of how much milk they can make. And so the benefit of the product, we're hypothesizing, manifested as a decrease in intake rather than a substantial increase in production. So some of the trials that are coming up are with individually penned animals, and so we'll be looking at that more over the next couple of months as well.
Ok, no sé si se entendió. Lo que cuento es que en los ensayos que han hecho en Estados Unidos, lo que se notó es que las vacas de alta producción, altísima producción, más que aumentar aún más ese nivel de producción, lo que hicieron fue bajar el nivel de consumo de materia seca, manteniendo ese alto nivel de producción. Así que lo que están haciendo es programando y diseñando nuevos ensayos para testear también esto o medir el consumo de materia seca.
Sí, doctor.
Bien.
Tengo una pregunta. Lo que es el país México, tenemos una gran producción de ganaderos, pero se utiliza en muchas unidades de producción lo que son productos para también elevar la producción de leche, digamos algún hormonal, por mencionar alguno. Le planteo la situación. Digamos que tengo producciones diarias por vaca de 50 litros. Aplicando esos productos que ya existen aquí en México, digamos que asciende a 52 litros diarios. La pregunta es, al agregar su producto Altius, ¿voy a obtener un incremento aún mayor?
Okay. So the question, Mallory, is when there are some products, and he asked specifically about hormones and growth promotants that are very common in Mexico. So, for example, if you have a 50 liters daily production and you use the hormones, you can go up to 52. So do you think that using Galaxies or Altius, you can even go higher than that?
Potentially. So we've tested Galaxis with other common additives, including rumensin, for example, and we found a stacking effect there. We have not tested it directly with hormones, though. So I can't say for sure if you'll see a huge effect if you stack it onto an animal that's already seeing a huge effect from the hormone. But they should not be interfering with each other, I guess is what I'm trying to say. They're acting in totally different ways. So you should see some stacking effect.
Okay. Sí, no sé si se entendió. Lo que dice es que puede haber una sumatoria de efectos, una apilación de efectos. No se probó específicamente usando hormonas en las vacas, pero sí se probó Galaxis o Altius usando promotores de crecimiento como por ejemplo la monensina, y sí hubo un efecto adicional. Entonces es de esperar que también haya un efecto adicional.
Ok, sí, queda respondida la pregunta, doctor. Gracias.
No, por nada. Si alguno tiene alguna otra pregunta para Mallory. Bueno, estamos entonces en tiempo. Si no hay más preguntas, vamos a terminar la reunión. Le agradecemos mucho a Mallory su participación y su apoyo, y obviamente a través de el equipo de ASCUS de Argentina y de México, si tienen algún otro tipo de inquietudes, alguna duda o quieren ver más información también sobre los ensayos que hemos venido haciendo desde el 2016, podemos también compartir toda esa información con ustedes. Mallory, we are going to close the meeting. A big thank you for your time and your very interesting and clear presentation. And we will keep in touch with folks here, with all our teams, to follow up on them and provide more information if they want to.
Great. Yeah, thank you guys for the attention.
Thank you, Mallory. Thank you, everybody, for joining the meeting.